Hi guys. So welcome back. Yeah. The paper that we are dealing with today is 2019 paper. Yeah. We have already seen 2020 paper. Let's go back a bit more and take conceptual based questions from 2019 paper. Right. So let's move ahead. Now, so question number 1. Respiratory quotient value of tripalmitin is. So let's first recall what is respiratory quotient. Yep. Respiratory quotient, we had studied respiratory quotient in respiration in higher plants, right? Respiration in higher plants. So there we studied what respiratory quotient is. So respiratory quotient is what? It is the ratio of amount of carbon dioxide evolved to the oxygen molecules consumed during the process, isn't it? That's how we define respiratory quotient, don't we? Right? So as we work on respiratory quotient, let us recall that carbohydrates. So respiratory quotient is the ratio of carbon dioxide evolved to the ratio of oxygen atoms consumed, isn't it? So let us recall that carbohydrates have a respiratory quotient of what? 1. What is the respiratory quotient for proteins? 0.9, isn't it? What is the respiratory quotient for fats? 0.7, correct? And respiratory quotient for anaerobic respiration, yep, it's infinity or it is undefined, isn't it? Yep. So tripalmitin is a fatty acid, isn't it? Yep. So the respiratory quotient here will become what? Oh, sorry, 0.7. Yeah, not 0.9. Yep, it's 0.7. Oh, by the way, you know, some things that I wanted to put forth are, congratulating our 10th ICSE students for scoring 100 on 100 in biology. Yeah. So good job, you people. Yeah. So next question. Which of the following is a commercial blood cholesterol lowering agent? Yeah. So when we talk about blood cholesterol lowering agent, which chapter are we talking about? We are looking at the source, isn't it? So this question is from Microbes in Human Welfare, where the blood cholesterol lowering agent is what? Statin. And what is the microorganism from where we get statin from? So Monascus purpureus, isn't it? Yeah. Next question. Match the following structures with their respective locations in organs. Now, so we have crypts of Leberkuhn. Where do we find crypts of Leberkuhn? Recall digestion and absorption. Yep, digestion and absorption. So crypts of Leberkuhn are pits which are found where? In the small intestine. So we have the villi, okay, and then we have these pits which act as crypts of Leberkuhn, don't they? Those are basically the crypts of Leberkuhn. And crypts of Leberkuhn also play an important role in immune system because they are also the house of what? They are also the house of Peyer's patches, isn't it? Yep. Next, Glysen's capsules, then we have islets of Langerhans, and then we have Brunner's glands, okay? So Glysen's capsules are the structures which are found where? They are found in the liver. Islets of Langerhans, we all know that islets of Langerhans are found where? They are found in the pancreas. Recall that islets of Langerhans have alpha, beta and gamma cells. Alpha cells secrete a hormone called glucagon. Beta cells secrete the hormone called insulin. And gamma cells secrete the hormone called somatostatin. Yep. So let's just like work out on the functions of these hormones also. So glucagon is what? Glucagon increases the blood sugar level whenever we are in starvation mode. Insulin decreases the blood glucose level, and somatostatin, somato body, soma body, statin stopping, isn't it? So somatostatin inhibits the secretions from alpha and beta cells, right? So islets of Langerhans, where do we have them? We have them in the pancreas. And Brunner's glands, where do we have Brunner's glands? We have Brunner's glands in the duodenum. They are responsible for secretion of certain hormones which further aid in the process of digestion, right? So that's basically the answer, isn't it? Yep. Is everyone following? Great. Which of the following muscular disorders is inherited? So let us take a look at all of these disorders one by one. Yep. Tetany. Tetany is a disorder that is caused due to the microorganism Clostridium tetani. Yeah. Clostridium tetani is the microorganism that causes tetanus or tetany. Yeah. It increases muscular spasms and it causes locked jaw. That is what is tetany. Muscular dystrophy is basically the answer over here. It's an inherited disorder. Yep, it's muscular dystrophy is an inherited disorder. It is inherited because of the problem with the dystrophin gene. Okay, myasthenia gravis. Myasthenia gravis is an autoimmune disorder. Yep, myasthenia gravis is what? It is an autoimmune disorder where the connections, the connections are basically broken down by our own body cells, our immune system. And botulism is another example of disease caused by a microorganism. Yep. So tetany, myasthenia gravis, botulism, these are the muscular disorders that are not inherited. Muscular dystrophy is the one which is an inherited disorder, right? Yep. Got it? Okay. Now, let's take a look at the next question. In a species, the weight of newborn ranges from 2 to 5 kg. 97% of the newborn with an average weight between 3 to 3.3 kg survive, whereas 99% of the infants born with weight from 2 to 2.5 kg or 4.5 to 5 kg die. Which type of selection process is taking place over here? Okay, so let us understand which type of selection process is taking place. For that reason, we need to go back to the chapter evolution. Correct? Now, we have directional, stabilizing, disruptive. These are the three types of selections that we see. So if we have a population, okay, if this is the population, then here we have what do we have over here? We have extreme values. Okay, extreme characteristics or extreme values. When one of the value or one of the phenotype is selected, when one of the phenotype is favored, then the curve will move in a particular direction. It will move towards one of the phenotypes, right? It will go like this. So this is what is directional selection, right? Then if there are two phenotypes that are preferred, then what will happen over here is we will see two peaks, and these two phenotypes will depend on where the organism is found. Okay, so the force of selection, the process of selection is occurring in this particular manner. So what is happening? We see two peaks. Whereas if an intermediate value is being selected, there is the direction of selection is occurring from both the sides, and a central value or a mid value or median value is what is being selected as a phenotypical characteristic. Are you understanding that? Yep. So this is what is stabilizing selection. Yeah. So this question. So let us understand it again. In a species, the weight of a newborn ranges from 2 to 5 kgs, right? 2 to 5 kg. So 2 kg is an extreme, 5 kg is another extreme. 97% of the newborn with an average weight between 3 to 3.5. So somewhere of the median value, 3 to 3.3 kg survive, whereas 99% of the infants born with weight from 2 to 2.5 kg, that is first extreme, or 4.5 to 5 kg, that is the next extreme, die. So which type of selection process is taking place? So the median value is being selected. So the selection is stabilizing selection, not disruptive. Okay. How does steroid hormone influence the cellular activities? Okay. So let's take a look at this. How do they influence the cellular activities? Recall that there are two type of hormones. Here the chapter is chemical coordination and integration, 11th graders. Right. So here what we are taking a look at is the activity of two hormones. Let's study them. So non-steroidal hormones and steroidal hormones. Okay. Now non-steroidal hormones are unable to cross the lipid membrane barrier, right? Isn't it? Because of the hydrophobic nature, they are unable to cross the barrier. So in non-steroidal hormones, what happens over here is we have a receptor. Okay. So for example, if this is the receptor that is present in the cell membrane. Okay. Right. Okay. So here we have our hormone. And then in the membrane what we have is the hormone receptor. Now when the hormone binds to the hormone receptor, what happens is a cascade mechanism is triggered, a cascade mechanism is created in the cytoplasm, right? And that cascade mechanism is cyclic AMP driven mechanism. And this cyclic AMP driven mechanism brings about the reaction. Like for example, if this hormone is glucagon, yep, it will go and bind to the liver cells. Why? Because in the liver cells we have glycogen stored, right? So glucagon will tell the liver cells to bring about glycogenolysis. What is glycogenolysis? It's the process of breakdown of glycogen, right? So it will bring about glycogenolysis, and because of this glycogenolysis, glucose will be secreted into the blood. Yeah, glucose will be secreted where? Glucose will be secreted into the blood. So this is what happens with non-steroidal hormones. Steroidal hormones, on the other hand, are capable of crossing the lipid bilayer. Okay, so if this is a steroidal hormone, like for example testosterone, progesterone, yep, estrogen, these are steroid hormones, isn't it? So they can simply cross the lipid barrier, okay, and move into the cytoplasm directly. Now there are two things that can happen. Every hormone requires a hormone receptor that can act as a transporter. So there can be a hormone receptor over here. Okay, this hormone receptor will bind to the hormone, the steroid hormone, and take it across the nucleus. Okay, and it will leave the hormone in the nucleus and it will come out to take another hormone. Okay. Now what will be the hormone doing when it is inside the nucleus? It will go and it will interact with the DNA at a locus called as the hormone receptor element, that is HRE. Okay, hormone receptor element on the DNA. And when it binds to the hormone receptor element in the DNA, what is going to happen is it is going to bring about a physiological action. So that is how a steroid hormone works. Okay, steroid hormone parent molecule is cholesterol, remember that. Yep. So answer will be what? Binding to DNA and forming a gene hormone complex, which will bring about a physiological action. Got it? Any doubt with this? Now, whatever doubts you have, put them in the comments. Yep, I'll take them. Next question. Consider the following statement. Coenzyme or metal ion that is tightly bound to enzyme protein is called prosthetic group. B, a complete catalytic active enzyme with its bound prosthetic group is called an apoenzyme. So let us recall cofactors for this, isn't it? Yeah. Now cofactors, we have prosthetic groups, coenzymes, and metal ions. Yeah. Now, what is a prosthetic group? A prosthetic group is a non-proteinaceous compound or a non-proteinaceous component which is tightly bound to the enzyme. Now, which enzyme? The proteinaceous part, the inactive proteinaceous part of the enzyme is called as apoenzyme. Okay, and when it's bound to the cofactor, what is it termed as? It is termed as what? Holoenzyme, isn't it? Yeah, it's termed as what? Holoenzyme. So the B statement is wrong automatically. Prosthetic groups are tightly bound, whereas coenzymes and metal ions may be transiently bound. That is, whenever it is required, they'll be utilized, or they will move out. That is what will happen with coenzymes and metal ions. But even though if coenzymes and metal ions are bound tightly to the apoenzyme to form the holoenzyme, they will still be considered as what? Prosthetic groups, right? So A is true and B is false, right? So here we have prosthetic group like heme, which is bound to hemoglobin, coenzymes like NADP, yep, FADP, yeah, nicotinamide adenine dinucleotide phosphate, flavin adenine dinucleotide phosphate, your favorite molecules. And then we have metal ions like Mg2 plus, which is responsible, which is one of the factors for restriction enzymes, which is one of the cofactors for hexokinase, which is an enzyme in the glycolysis pathway. Correct? So that was about cofactors. Let us move ahead with the next question. Select the correct sequence for transport of sperm cells in the male reproductive system. So when the sperms are transferring from the testes to the penis, or their removal from the penis, so it will be moving from where? It will be moving from seminiferous tubules to the urethral meatus. What is the urethral meatus? Please recall that urethral meatus is the opening of the urethra on the head of the penis, right? So seminiferous tubules to the urethral meatus. So we have to take a look at the pathway between these two, the seminiferous tubules and the urethral meatus. Yep. So seminiferous tubules to urethral meatus, which options are giving us that, which of the options are giving us that, those terms? I can see seminiferous tubules in B. I can see urethral meatus in D. Okay. But testes, epididymis, no. So this here is the correct option. So from the seminiferous tubules, recall that in the testes, so if we look at the testicle, okay, we have these lobes, yeah, and inside these lobes we have highly coiled seminiferous tubules, correct? And these highly coiled seminiferous tubules, they form a reticulated network called the rete testis. And from this rete testis, we have certain efferent ducts. What does efferent mean? Efferent means going out, isn't it? efferent ducts. So that is vasa efferentia, isn't it? Vasa efferentia. These efferent ducts, they open into the epididymis. Epi, above. Didymos means testicle, right? So epididymis is the structure which is a highly coiled structure present on what? Present on the testicle, right? So we have the head of the epididymis, which is caput epididymis, corpus epididymis, and then we have the cauda or the tail epididymis, the cauda epididymis. Now from the cauda epididymis, the tail epididymis, what arises and loops out is the vas deferens, correct? When the vas deferens loops over the ureter and joins the urethra, it forms the ejaculatory duct, right? Yeah. And then it continues as the urethra and ultimately opens into the urethral meatus. Got it? Yep. So seminiferous tubules, rete testis, vasa efferentia, epididymis, vas deferens, ejaculatory duct, urethra, and urethral meatus. This is the sequence of the passage of sperms from the seminiferous tubules to the outside of the body. Got it? Yep. Wonderful. Nice question. What would be the heart rate of a person if the cardiac output is 5 liters? Blood volume in the ventricles at the end of diastole is 100 ml and at the end of ventricular systole is 50 ml. Cardiac output. Cardiac output equals to the stroke volume multiplied by the heart rate. Yeah, correct. So we have cardiac output which is 5 liters, yep, which we can simply make it, convert it to 5,000 ml, right? Stroke volume and heart rate. Now they are asking us the heart rate, so we are supposed to find out the heart rate. But have they given us a stroke volume? They have given us the stroke volume, but they haven't stated it explicitly. So let us understand. Blood volume in the ventricles at the end of diastole is 100 ml. At the end, so there's diastole, systole, diastole. So if at the end of diastole the blood volume is 100 ml and at the end of ventricular systole it is 50 ml. Where is this? In the ventricles. That means the ventricles have pushed out how much amount of blood? 100 minus 50, isn't it? So the stroke volume for this athlete is 50 ml. And what are we supposed to find? The heart rate. So do the math. Yeah. So how much do we get it? 100 beats per minute. I hope everyone has understood this. Yep. How we got the stroke volume as 50 ml. Which of the following factors is responsible for the formation of concentrated urine? Okay. Urine concentration. What is the mechanism? The mechanism that is utilized is countercurrent mechanism, isn't it? Which is the mechanism of concentration of urine, right? Why does the countercurrent mechanism work? Because the movement of the glomerular filtrate through the nephron, okay, as it moves through the proximal convoluted tubule into the loop of Henle. So we have the proximal convoluted tubule over here, then the loop of Henle, right? and then we have the DCT, correct? Yeah, and finally the collecting duct. So this is coming from where? This is coming from cortex to the medulla. So as the loop of Henle is dropping down from the cortex to the medulla and rising back into the cortex from the medulla, we see that it has descending loop and ascending loop, descending limb of loop of Henle and ascending limb of loop of Henle. So what happens over here is, around this, what do we have? We have the vasa recta, isn't it? And the movement of blood in vasa recta is opposite excuse me, opposite of that of the movement of glomerular filtrate in the nephron. So as the glomerular filtrate is moving downwards and upwards, the blood is moving in this direction. So glomerular filtrate is going down over here and up over here. The blood is moving down over here and up over here. And this countercurrent mechanism establishes something which is called as hyperosmolarity. Yep. So where does it maintain the hyperosmolarity? It maintains the hyperosmolarity from the cortex to the medulla. What do you mean by osmolarity? It's another method of telling you concentration. So it's dependent on the number of solute particles. So if higher is the osmolarity, then the concentration of the solution is high. So if the medullary interstitium has high osmolarity, yep, hyperosmolarity towards the inner medullary interstitium, that means what? The medullary interstitium will be hypertonic, wouldn't it be? Yeah. So since the medullary interstitium is hypertonic and the descending limb of loop of Henle is permeable to water, water can move out readily from the descending limb of loop of Henle into the medullary interstitium, and it is immediately removed from the medullary interstitium to maintain the hyperosmolarity so that water reabsorption continues throughout the tubule. Are you getting it? Yep. So these are the factors. This is how countercurrent mechanism works, and it is responsible for the concentration of urine. Got it? Which of the following contraceptive methods involve the role of hormone? Right. So let us just revise the methods of contraception. Yeah. So contraceptive methods, natural and artificial. Right? Contraceptive methods, we have natural and artificial. Natural is complete abstinence till the time you are ready to bear children. Periodic abstinence, which is the rhythm method, that is not having sexual intercourse during the fertile period of the menstrual cycle. We have coitus interruptus, or the withdrawal method. And then what we have is lactational amenorrhea. Yeah. Lactational amenorrhea. Amenorrhea means what? The absence of menses. Menorrhea. So menstrual cycle is not occurring. Amenorrhea, menstrual cycle is not occurring. Why? Because the mother is undergoing lactation. Are you getting it? So lactational amenorrhea is the condition wherein, because of the hormonal conditions, the menstrual cycle is not occurring. But this is maximum validity of this is about 6 months. Beyond that, it fails. Okay. Artificial, we have temporary methods and permanent methods, right? Under temporary methods, we have barrier methods, okay? Then we have intrauterine devices. We have chemical methods like pills. These can be morning after pills or emergency pills. And permanent methods will be what? Surgical, isn't it? Vasectomy, tubectomy. Correct? Vasectomy, tubectomy. So barriers are going to use what? Condoms, femidims, and diaphragms or cervical caps. IUDs are intrauterine devices, copper teas, Lippes loop. Yep. Pills will again have hormones in them, isn't it? These are progesterone pills. Yep. Why do you think progesterone pills are going to work? Understand this. This is where the hormonal control of menstrual cycle comes into picture. Progesterone provides a negative feedback on LH and FSH. Since there is a negative feedback on FSH, follicular phase of the menstrual cycle cannot occur. Since the follicular phase of the menstrual cycle is not occurring, more follicles are not going to develop, and neither an ovum is going to be maturing. So there is no estrogen, okay? There is no LH, so there's not going to be any ovulation. Progesterone keeps or maintains the lining of the endometrium. So because of which menstruation is not going to occur. So progesterone is preventing menstruation and also providing negative feedback on LH and FH. Yep, so it is preventing the occurrence of the menstrual cycle in this particular manner. So if the menstrual cycle is not occurring, will there be any pregnancy? No. So the answer over here is what? Lactational amenorrhea, pills, emergency contraceptives. Got it? Next question. Tidal volume and expiratory reserve volume of an athlete is 500 ml and 1000 ml respectively. What will be his expiratory capacity if the residual volume is 1200 ml? Understand the question. Residual volume over here has no significance because residual volume remains undisturbed, right? They are asking for expiratory capacity. So expiratory capacity is what? It's the amount of air that you can breathe out by forceful expiration. So that will comprise of what? Tidal volume and your expiratory reserve volume. So tidal volume, they have already given us 500 ml, which is the regular case, and expiratory reserve volume 1000 ml. So what's the expiratory capacity? Got it? So don't get thrown off by residual volume. Understand? Remember, the residual volume is the amount of air that is always present in the lungs. No issues, right? It's never coming out. Following statements describe the characteristics of the enzyme restriction endonuclease. Identify the correct statement. The enzyme cuts DNA molecule at identified position within the DNA. Incorrect statement. We have to take a look at. The enzyme binds at specific sites and cuts only one of the two strands. The enzyme cuts the sugar phosphate backbone at specific sites on each strand. The enzyme recognizes a specific palindromic nucleotide sequence in the DNA. Yep. So let's take a look at it. What are we talking about? We are talking about restriction enzymes, and most importantly, restriction sites or recognition sequences. Recall that recognition sequences are small 4 to 8 nucleotide sequences, yep, which are palindromic in nature. Yeah, palindromic in nature. Now, what do you mean by palindromes? Palindromes are the words that can be read same in both the directions. Level, dad, mom, Malayalam, all of them. Got it? So palindromic in nature. So for example, we have an enzyme called EcoR1, restriction enzyme, which is obtained from Escherichia coli. So it recognizes a sequence GAATTC 5' to 3', and same thing on the opposite side, 5' to 3', GAATTC. Right? So this is the recognition site for EcoR1. Remember, restriction enzymes have their own specific recognition sequences or restriction sites. Yeah, EcoR1 is not going to cut the restriction site of BAMH1 or HIN2. They all have their own recognition sequences. This is a palindromic sequence. Restriction enzymes that are used in recombinant DNA technology make a cut within the site. Okay, and it is highly specific, and both the strands are cut simultaneously. So the option over here that says cuts only one of the two strands is the incorrect one, isn't it? Yeah. All other options satisfy the function or the process with which restriction enzymes bring about the cleavage of the DNA. Yep. Remember that restriction enzymes bring about the cleavage of the DNA by hydrolysis of the phosphodiester bond. Yep, hydrolysis of the phosphodiester bond. Next, another question from respiration. Conversion of glucose to glucose 6-phosphate. The first irreversible reaction of glycolysis is catalyzed by aldolase, hexokinase, enolase, phosphofructokinase. Yep. If now you understand why enzymes, remembering enzymes, understanding enzymes is important. Glycolysis begins with glucose, isn't it? It's the process of breakdown of glucose, glycolysis. Glucose is converted to glucose 6-phosphate, right? So there is phosphorylation. Now, who is the phosphate donor over here? So the phosphate donor is ATP. Yep. And the enzymes that are responsible for transfer of phosphate to and from ATP are kinases, right? So the answer over here is hexokinase. Why hexo? Because glucose is a 6-carbon sugar. It is a hexose sugar. And recall that the enzymes are named based on the substrate they act upon. So hexokinase. So it is acting on a hexose sugar, and hence it is called as what? Hexokinase. Yep. And it requires Mg2 plus as cofactor. Got it? Yeah? Wonderful. Oh, good. A gene locus has 2 alleles, capital A, small a. If the frequency of the dominant allele A is 0.4, then what will be the frequency of the homozygous dominant, heterozygous and homozygous recessive individuals in the population? What are we dealing with over here? Hardy-Weinberg principle from evolution. So Hardy-Weinberg principle states what? If the frequencies of the alleles are not changing, or frequency of the allele should be constant, If they are not, evolution is taking place. Okay, so Hardy-Weinberg principle can be given by a simple formula: P plus Q the whole square equals to 1. Yep. So it's expected to remain constant. Yep. Or P plus Q equals to 1. Correct. Now, what does P stand for? P denotes the dominant allele, Q denotes the recessive allele. And if we open the brackets, it is P square plus 2PQ plus Q square, right? P square gives us the frequency of the homozygous dominant condition. 2PQ gives us the frequency of the heterozygous condition. Q square gives us the frequency of the homozygous recessive condition. Yep. So what are we actually provided with? The frequency of the dominant allele. That is 0.4. So we are given P. We have to find Q, isn't it? So P plus Q equals to 1. So Q is going to be how much? Right. Simple. Yeah. Now, if I want, now what are they asking us? What will be homozygous dominant, heterozygous, and homozygous recessive? So P square will be how much? 0.4 square. This will be how much? 2 into 0.4 into 0.6. And this is going to be 0.6 square. So 0.16, 24 into 2, 0.48, 6.36. Right? Wonderful. So frequency. Got it? Hardy-Weinberg principle. Simple enough? Yep. Great. Let's move to the next question. Which of the following is true for golden rice? Golden rice is a genetically modified rice. Yeah. What is it genetically modified for? It is a rice which is enriched with provitamin A or beta carotene to overcome vitamin A deficiency in people. It's a good example of biofortification by genetic modification. What is biofortification? It's increasing the inherent quantity of nutrients in the source of food. That is what is biofortification, biological, fortifying, strengthening, right? So biofortification. Provitamin A, beta carotene. Okay. The sources for this particular gene to make it golden are two organisms, Erwinia euredivora, which is a microorganism, and the other is a plant called Narcissus pseudonarcissus. Narcissus pseudonarcissus is commonly called as daffodil. Yep. Got it? So vitamin A enriched with a gene from daffodil. Erwinia euredivora and Narcissus pseudonarcissus are the two source of organisms from where you get the extra genes for golden rice. Expressed sequence tags refer to genes expressed as RNA, polypeptide expression, DNA polymorphism, novel DNA sequences. Where did we study express sequence tags? Right? Human Genome Project, isn't it? Human Genome Project what are express sequence tag? Let the term expressed sequences guide you. When we say expressed sequences, that means what? There is a gene sequence, a cistron, that has undergone gene expression, isn't it? Yeah, a cistron that has undergone gene expression. So if a cistron has undergone gene expression, what are we going to get? We are going to get a corresponding mRNA. We are going to get a corresponding protein. In express sequence tag, what is focused upon is the mRNA. Yeah, and recall that eukaryotic systems have something which is called as post-transcriptional modifications. Yeah, and these post-transcriptional modifications have what? Exon splicing, 5' capping, and 3' polyadenation. So because of these 3 post-transcriptional modifications, do you think that if you look at the mRNA sequence, you're going to get the exact complementary DNA sequence? That's not possible, because the introns have been removed, and these introns were a part of the original DNA sequence, isn't it? Yeah. So mRNA is subjected to reverse transcription, and what is obtained from that is a DNA sequence which is known as cDNA, or complementary DNA, which is again made double-stranded with the help of DNA polymerase and inserted into the plasmids, which are further inserted into bacteria and cloned and stored. Okay, so expressed sequence tags are genes expressed as RNA. Got it? Which of the following immune responses is responsible for rejection of a kidney graft? So if a person receives a kidney, you have heard that organ has been rejected, etc. All of these things happen. Now why does that happen? Is because on the cells, on the organs, see, we are dealing with tissues. Yeah. So over here we are not dealing with hemocompatibility. We are not dealing with blood-blood compatibility. We are dealing with tissue compatibility. So it is called as histocompatibility. Yeah, and histocompatibility is controlled by something which are called as major histocompatibility complexes, MHCs. Yeah, MHCs, which are present on the tissue cells and the cells of the immune system. So if these MHCs, that is major histocompatibility molecules, do not match, then there will be the rejection of the tissue graft. But the immune system, but the immunity that will always be responsible for that is cell mediated, isn't it? Because we are talking about specific response, and specific response is always cell mediated immunity. Autoimmune response is a disorder. Humoral immunity is a collective term used for what? Blood related immunity. Yep. So everything related to blood, see, humors is a common term for liquids in the body. Yeah. So lymph, blood, your aqueous humor, vitreous humor, liquids from the body, isn't it? Yeah. So humoral response. So any response with respect to lymph or blood will be termed as a humoral response. It's an umbrella term. Inflammatory immune response is what? It's inflammation. It is the response of the first order, isn't it? Yeah. Non-specific immune response. But specific immune response, where a graft is being rejected, is cell mediated. Match the following genes of the lac-operon with their respective products. Great. I think this is easy. You can pull it off very easily, isn't it? Yeah. Let's just understand the functions. So beta-galactosidase is responsible for what? Beta-galactosidase is the enzyme that is responsible for hydrolysis of lactose to glucose and galactose, right? Permease is responsible for what? Entry of lactose in the cell. Repressor binds the operator and prevents transcription of the lac-operon genes. And transacetylase, what is it responsible for? Transacetylase is responsible for transfer of acetyl group. Transacetylase, it's transferring acetyl group to beta-galactosidase so that it can perform its function of hydrolyzing lactose into glucose and galactose. So I gene is the one that gives us the repressor, Z gene is beta-galactosidase, A gene is transacetylase, and Y gene is the permease. Got it? Yeah. Under which of the following conditions will there be no change in the reading frame of following mRNA? Recall what are frame shift mutations, guys. Frame shift mutations are the mutations which occur due to deletion or addition. So if it says insertion of G at 5th position, there is only one nucleotide being added. So obviously there will be frame shift. Deletion of G from 5th position, single nucleotide is being deleted, there will be frame shift mutation. So the reading frame will shift. Insertion of A and G at 4th and 5th positions, 2 nucleotides are being added. Okay, recall that a codon has 3 nucleotide sequence, 3 nucleotides. So if 2 are added or even if 2 are deleted, the reading frame is still going to change. So what does it say? Deletion of GGU from 7th, 8th and 9th position. So if 7th, 8th and 9th position GGU is deleted, an entire codon is gone. So since an entire codon is gone, that particular amino acid which was signified by GGU is also gone. But the reading frame of the other amino acids, the reading frame of the other codons on the mRNA is intact. Yeah. So which of the following conditions will there be no change in the reading frame? Deletion of GGU from 7th, 8th, 9th position. Yep. What triggers activation of prototoxin to active Bt toxin of Bacillus thuringiensis in bollworm? Simple. Prototoxin comes in contact with alkaline pH of the gut. And the activated prototoxin, what does it do? The activated prototoxin brings about the destruction of the epithelial lining of the gut of the insect. It creates pores in the lining of the gut of the insect. And ultimately what happens over there is inflammation or swelling in the gut of the insect, because of which the insect is unable to eat anymore and it dies of starvation. Got it? Yep. So alkaline pH of the gut. And the last question for today. What is the genetic disorder in which an individual has an overall masculine development, gynecomastia, and is sterile? Okay, you all know this is Klinefelter syndrome, correct? Let's just take a look at the genetic condition. So Turner syndrome is what? 44 plus XO, absence of one sex chromosome. So this is monosomy, isn't it? Klinefelter's is 44 plus XXY, yeah, trisomy of sex chromosomes. Down syndrome is trisomy of 21st syndrome, isn't it? Yeah, got it? Edwards syndrome is not in your syllabus, so don't bother about it. Yep. So 44 plus XO, 44 plus XXY, and trisomy of 21st chromosome, Down syndrome. Got it? So these are the genetic conditions. Yep. So that was all for today. Okay. I shall see you next time with another paper, and we will keep on discussing this. Yep. So have a good day, guys, and keep studying.